10 seconds to just make their way to their seats, then we can proceed. I hope everyone enjoyed the poster session. There's also posters way at the back. It was so sad. The people who were standing that side were very lonely. So please, during the poster session, please also just move across and see them as well. Okay, so we will officially start. So good morning, colleagues, and welcome to the second session of the CSIR Emerging Research Symposium. And my name is Sindisiwe Buthelezi. I will be chairing this session focused on health and medical science, a field that continues to drive innovation towards diagnostics, therapeutics, and public health. So this session we will be hearing from 5 promising researchers and who are playing an active role in shaping the future of our health in South Africa and beyond. So before we start, I would like to just do a few housekeeping notices. So number 1, I've been tasked to just remind everyone to please register on the actual system as they are collecting the statistics and the data. So not just the manual registration, but please also find someone, think they're at the front desk, to help you enter your details in the app as well. And then please ensure that your devices are on silent, and the speakers will all get 15 minutes to give their presentation. Then there'll be 5 minutes for Q&A. And then to the speakers, when you have 5 minutes left, I'll just show my paper of 5 minutes. Then when you have 1 minute left to wrap up, I'll just stand on the side. All right, so please help me welcome our first presenter, who is Sesianda Masego. The title of her talk is Whole Genome Characterization of Noroviruses Detected in Children Under 5 years from Gauteng, South Africa. Thank you.
Good morning, everyone. My name is Sesianda Masego, and today I will be presenting my preliminary findings for my master's project. Oh, my fault. Oh. Yes, so as I said, today I will be presenting my preliminary findings for my master's project. It is well documented that noroviruses are among the major diarrheal causes. But what if other enteric viruses are silently present, contributing to the overall disease burden? To explore this, metagenomic next-generation sequencing of norovirus positive fecal samples was conducted, which was obtained from South African children under 5 years, and this revealed norovirus diversity and co-infections with other enteric viruses. Diarrheal diseases are among the major causes of infectious disease morbidity and the 14th leading cause of mortality across all age groups. In children under 5 years, unfortunately, the impact is even more severe, with it ranking the 3rd leading cause of mortality. Viruses are the common cause of acute gastroenteritis, including rotavirus, norovirus, sapo, and astroviruses and adenoviruses. Among these, noroviruses have been recognized as the leading cause of acute gastroenteritis across all age groups. And this has been demonstrated by its significant global disease burden, with an estimated 685 million infections reported annually. Children under 5 years are particularly vulnerable, with over 200 million infections and 95,000 deaths reported annually. In sub-Saharan Africa, norovirus accounts for 19% of global infections, with South Africa having a prevalence of between 20 and 30%. We now know that noroviruses exert a significant disease burden, but what exactly are noroviruses? Noroviruses are single-stranded RNA viruses. They are classified within the Calliciviridae family. They have a non-enveloped structure with the VP1 major capsid protein forming an external shell, encapsulating a genome of approximately 7.7 kilobase pairs, which consists of 3 open reading frames. They are a genetically diverse group of viruses divided into 10 genogroups, which are further subdivided into 49 genotypes and 60 polymerase types. Although noroviruses exhibit a significant diversity, human infections are mainly caused by genogroups 1, genogroups 2, 4, 8, and 9. Despite norovirus' significant disease impact, there are currently no vaccines or treatments for noroviruses, with challenges including high genetic diversity and the lack of cross-protective immunity. A key approach in advancing norovirus surveillance is the genomic surveillance, which enables strain tracking and thus informs vaccine design. Although partial sequencing methods have provided valuable information, they lack the resolution required to capture full viral diversity. Whole genome sequencing via next-generation sequencing, on the other hand, offers high resolution and thus detailed genetic insights. Moreover, metagenomic next-generation sequencing approaches, which use non-specific primers, provide an even broader advantage as they allow for the detection of novel and co-infecting viruses, which would have been otherwise missed by using amplicon sequencing. Despite the advantages that these studies offer, such studies are scarce in Africa, even though the continent bears a significant disease impact. And to then address this gap, this study aimed to characterize norovirus genetic diversity using whole genome sequencing and to detect co-infections with multiple norovirus strains and co-infections with other enteric viruses using MNGS. This study forms part of a larger study, the CSIR. Ethical approval was obtained from the UFS-HS-REC and UFS-EB-REC. For this study, a total of 84 archived norovirus-positive fecal samples from children under 5 years old from the Gauteng province, South Africa, between 2008 and 2019 were retrieved from the NICD. To then process these samples, the NETO-VIR protocol was then followed, where viral enrichment was firstly conducted following the necessary steps, followed by nucleic acid extraction using the respective kit, then cDNA synthesis and whole transcriptome amplification, followed by library preparations, and lastly, next-generation sequencing using the NextSeq 2000. The Illumina raw reads were then processed using 2 web-based bioinformatics tools, and those were Genome Detective and Trans-Logopic ID, where low-quality reads were filtered, adapters trimmed, and reads assembled into context, as well as viral sequences identified. The human calicivirus typing tool, as well as the norovirus typing tool, was then used to assign norovirus genogroups, genotypes, as well as polymerase groups and types. To give you an overview of the metagenomic data, 84 norovirus positive fecal samples were processed, of which 55 norovirus full genomes were generated and 2 near full genomes, and with the remaining 27 samples either producing partial sequences or having a few reads to proceed with further analysis. Over 12 million mapped reads were generated, with 91% of those belonging to noroviruses and 9% to other co-infecting viruses. To give you a perspective of my preliminary findings, we will firstly focus on noroviruses. This pie chart depicts the distribution of norovirus genotypes, and evidently G24 was the most predominantly detected strain, followed by G26 as well, and G23 and other norovirus genotypes circulating at lower levels. This bar graph depicts the yearly distribution of norovirus G24 variants, and evidently it indicates that G24 was not a single static strain, but rather several G24 variants, namely Allegheny, Appledoorn, New Orleans, Osaka, and Sydney, appeared and replaced each other over the years. A very interesting observation was the detection of the G24 Sydney variant in 2009 and in 2010, which was way before the major 2012 Sydney outbreaks. This study also observed a recombination frequency of 73.7%, and a recombination frequency of 73.7%, and this was indicated by mismatching in the capsid and the polymerase, and this assisted in giving an initial insight into which recombinant strains would have been circulating during this period. Additionally, 9.5 recombinants were collected in the same year and same site. For instance, in 2011, 2 G26P7 strains were collected from site 2, and in 2017, 2 G24 Allegheny P31 strains were collected from site 10. Still on noroviruses, this study observed a co-infection frequency of 21%, with samples containing up to 3 different norovirus strains. For instance, patients with sample number 8215 was co-infected with a G2 strain, a G4 strain, and a G1 strain, G4 strain, and a G24 strain. Now that we've looked at noroviruses in detail, let's now shift our focus to co-infections with enteric viruses, so other enteric viruses. Evidently, rotaviruses were the most commonly detected, which co-infected 35 out of 57 patients, followed by adenoviruses, then Astro, and lastly Sapo. So the darker shade of red indicates a high viral read count, and evidently rotaviruses had a higher viral read count compared to the other viruses. Beyond co-infections with common enteric viruses, the study also detected co-infections with other enteric viruses, and evidently enteroviruses and pericoviruses were the most frequently detected, with other viruses detected at a lower frequency. And as with the previous heat map, the darker shade of red indicates higher viral read count, and enteroviruses and pericoviruses accounted for most reads. Evidently, metagenomics enabled us to recover norovirus genomes as well as to detect other enteric viruses. And the partial, or the 65.5 recovery of whole genomes was mainly due to there being a few viral reads. And this basically highlights that in as much as metagenomics enables us to capture other enteric viruses, these may be more abundantly captured than the intended target. Despite the partial recovery rates, the recovered genomes did reveal norovirus genetic diversity, with G24 being the most dominant, followed by G23 and G26. So the G24 dominance could be explained by the emergence of multiple G24 strains, which could have escaped acquired immunity. An interesting observation, as I have mentioned, was the detection of the G24 Sydney variant in 2019 and in 2010, which was before the major 2012 Sydney outbreaks. And this basically highlights the importance of doing early genomic surveillance, as this will enable us to detect these strains before they become more widespread and thus help us to prepare for future outbreaks accordingly. The high recombination frequency that was observed in the study reinforces what has been widely reported, as it has been reported as a major driving force of norovirus diversity. The 9.5% recombinants that were collected in the same year and site could imply that they're from a shared source. However, transmission chains have to be done to basically determine or indicate whether these infections were indeed linked or independent. Building onto norovirus diversity, the fact that 21 patients were infected with more than one norovirus strain basically indicates the genetic diversity that can occur in just a single infection. And this is particularly concerning, as studies have reported that norovirus co-infections facilitate recombination and thus the emergence of novel variants. The high occurrence of co-infections with enteric viruses could be explained by poor sanitation and unsafe water in the setting, as well as the fact that these pathogens share a fecal-oral route transmission, and thus increased susceptibility may occur after the primary infection. Rotaviruses being the most commonly detected is consistent with global studies, and this is explained by rotaviruses being the leading cause of age in children under 5 years. The fact that high occurrence of co-infections, single enteric infections cause significant morbidity. The fact that high occurrence co-infections were detected in this study and multiple pathogens may infect just a single host, this could lead to increased morbidity, complications, as well as mortality, and this has been explained mostly in children in LMICs. In conclusion, the preliminary findings of this study provide insights for vaccine developments by basically demonstrating both emerging as well as dominant strains. The predominance of G24, which is consistent with global and local findings, reinforces its ongoing role in norovirus infections, and it makes it an attractive target for vaccine development. However, the genetic variability that was observed may limit strain-specific vaccine efficacy, and thus is a need for a cross-protective vaccine. The high occurrence of co-infections that was observed in the study indicates that other enteric viruses are indeed silently present, which may complicate control efforts and thus make treatments less efficient. This then emphasizes the need for a broad multi-pathogen metagenomic surveillance beyond single virus testing. Now that the preliminary findings for this study are now in place, the next steps are to ensure completion are to determine recombination breakpoints, phylogenetic analysis, elucidating transmission patterns, and this will assist in building a more comprehensive understanding of noroviruses and thus guide future responses as well as prevention strategies. I would like to acknowledge the following. Thank you. Stool samples, fecal samples. Yes.
Any questions?
How big was the sample or the number of feces that you'd got from the patients? How many patients?
Oh, it was 84. Yes.
From one area or from different provinces?
It was from Gauteng. It was different areas in Gauteng.
Any more questions? Maybe another one from myself while the colleagues are thinking is the co-infection, the viruses, is there one now that you always expect to be there? And does it somehow interact now with the norovirus? Is there a particular reason why it always seems to go hand in hand or with the norovirus infections?
Sorry, can you please say that again?
So the viruses that are co-infecting, are there like particular viruses that you would expect? So if you see the norovirus, you know that the other virus will be there as well. And if that's the case, is there any maybe interaction between these viruses that will that encourages the co-infection?
Not necessarily. So there's no specific virus that is expected to be always co-infecting together with noroviruses. It usually interchanges. Yeah, so there's no specific one.
Is it region-based or?
I think region would or can play a role, because especially because noroviruses are transmitted by unsafe water, etc. So basically, if a region is to have unsafe water, then there could be a high chance of co-infections occurring in that area.
Thank you. Any more questions? Okay, thank you.
Thank you.
So now I'm not sure. All right. Our second speaker is Oyomide. I hope I'm not mispronouncing your name, Abe, whose work lies in the intersection of medical imaging, artificial intelligence, and diagnostic innovation. So the title of the presentation is Lung Cancer Diagnosis from Computed Tomography Scans Using Deep Learning Algorithms and Marvage Pooling and Random Pixel Swap Data Augmentation Technique. Thank you so much.
Good morning. I'm happy to be here. So I'll be presenting results from my PhD studies. It was supervised by Professor Nyati, Professor Okwande, and Professor Piloy. The title of my presentation is Lung Cancer Diagnosis from Computed Tomography Scans Using Deep Learning Algorithm with Marvage Pooling and Random Pixel Swap Data Augmentation. Here is my presentation outline. Lung cancer is the most common cancer for all genders. It is the highest cancer-related deaths in the world. The average, 5-year survival rate for lung cancer is very low compared to other cancer types, which makes detection of lung cancer to be a critical need. For example, if lung cancer detected early, 55% of patients survive more than 5 years compared to when it's detected late, where we can see about less than 5%, who survive. In South Africa, about 8,000 lung cancer cases were diagnosed between 2017 and 2019. In 2020, the value rose to 8,950, which represents the highest case in a single year. To achieve early detection, automated systems have been proposed, particularly deep learning. It has shown to surpass the capabilities of radiologists, but they encounter a lot of challenges. For example, high computational costs, which affects, training and then using resource-constrained settings. Also, the challenge of data where to get training data becomes very difficult because it requires high expertise annotation, and also you need large volumes of data to train the algorithms. For our study, we aim to achieve a early detection whereby we can design a system that is less computationally intensive, and then we can train that on a very small amount of data using, for example, a case hospital scenario, and then we can also achieve results that are similar to oncologists, to radiologists, or even better than results from radiologists. Basically, we are not designing to, take the jobs of the radiologists, but at least to, give them informations quickly about the analysis. To perform our experiment, we designed several, methods, which some of them are novel, and then we compare our methods with existing architectures. For example, AlexNet, GoogleNet, ResNet, MobileNet, and also some transformer architectures such as a Vision transformer and the Swing transformer. For the novel technique we developed, the Mavid pooling, we compare with the standard pooling techniques, which are the max pooling and the average pooling techniques. The experimental setup, we use a PyTorch running on a Jupyter notebook, and then we train the system using an NVIDIA GPU. We use the ADAMW optimizer. We use cross-entropy loss function. We use the step LR scheduler, and most cases we used a batch size of 16. For our statistical analysis, we evaluated the model on accuracy, specificity, sensitivity, and area under the receiver, operating character receiver curve. We also performed PIT tests to test significant difference. We developed a novel metric, which is the cumulative scores, given by that equation to tell us, for example, if we have 3 different models or 3 different techniques, which is the better technique, over several experiments. The first method that we proposed was the random pixel swap data augmentation, which we published in this journal. What does it do, basically? Because we know that unlike conventional images where you can go to Google, for example, if you want images of cats, it's easy to get millions of images of cats, but when it comes to medical imaging, it becomes very difficult because of the high specificity that is required and accuracy to develop models like that. So because of this, data is very scarce for training lung cancer models. So we developed a method that can augment the data, which we call the random pixel swap data augmentation. What it does is that it's a patch-based technique whereby if you have an input image, for example, this image, we can then transform this image to provide meaningful representation of the data, and then we can achieve multiple images to train the model. Why we developed this was because available techniques, they fail particularly for, when you're using them for maybe CNN and transformer architectures. They do not generalize to both architecture paradigms. And also, unlike conventional training, if you're training a classical or conventional model, these techniques, they perform well. But when it comes to medical imaging, they fail, or in fact, they underperform and limit performance. So here is the algorithm that we developed for the random pixel swap data augmentation technique. Also, we compare it to state-of-the-art techniques such as cat mix, random erasing, mix-up, and cutout. These are standard techniques in literature. We evaluated the average training time, and then we saw that although our technique showed the largest training time, but this was not significant compared to other techniques. The difference was not significant. This makes our technique to be on par with the standard techniques in terms of training times. We also evaluated a technique for lung cancer detection, where we saw that our technique performed better than other techniques available in literature, which are the state of the art. And using our cumulative rank score, we saw that our technique ranked the best, as you can see, rank 1 here, for this particular task in detecting lung cancer. Also, we tested that on lung cancer classification. It also saw a similar performance where our technique, the random PJ swab, surpassed almost all the techniques considering the metrics. And this difference were actually significant. It is customary to train algorithms using segmented images because it has been shown that these images, if you segment the image, they can improve performance. So we tested to see if this technique used with segmented images can improve performance. And the result is presented here, where we see that our technique surpassed almost all the techniques, and it consistently ranked number 1 compared to the state-of-the-art techniques. Also, there are available traditional data augmentation techniques, such as flipping and rotation. We also check that if we combine our technique with this traditional augmentation technique, does it improve the performance, or does it perform better than using this technique? In most of the cases, we see that our technique alone, surpasses the performance of the traditional techniques. And also, when we combine the traditional technique with our technique, in some cases, it improves the technique, but in some cases, our technique alone surpasses, combining these techniques with our technique. Reason because some of these traditional techniques, in fact, they reduce the performance of the model. So when you now incorporate our technique, it struggles, to improve performance due to the, loss in performance of the traditional techniques. Also, we validated our experiment because one of the problems of deep learning is if you train a model on the data that it is used to, for example, let's say George Mukhari Hospital in Sefako Makato, you use the data there, you train the model on it, and then you go to Steve Biko Academic Hospital, and then you try to implement the model to analyze same CT scan data, you see that it fails drastically and woefully. So we try to see that can our technique actually improve generation capabilities. So we take data from different regions and different city protocols. We trained on one and then we tested on the other. We saw that our technique, in fact, performed excellently compared to other techniques, and in fact, it ranked number 1 for all metrics with significant difference. The second technique that we propose is the average pooling technique. What does this technique do? Remember, we want to develop an algorithm that is computationally effective, and then we can train it on relatively small amount of data, for example, that can be used in the clinical local hospital settings. In CNN architecture particularly, it has different layers, so we tackle the pooling layer. So what pooling does is that it selects, it downsamples the image through different mechanisms. The major ones that are available in literature, which are very common, that are used in architecture, such as the GoogleNet and the ResNet, is the max pooling and the average pooling techniques. But we see that when we apply these to CT images, when the model gets deeper, it loses signals. Sometimes the model overfits the data. So we come up with this technique to replace the pooling layer in the CNN and then with the average pooling technique. Basically, this is what the average pooling does. If you have an input feature, for example, in here, you can see if you apply a 2x2 max pool, you get this value. If you apply a 2x2 average pooling, you get this. But if you apply our technique, which is the 2x2 average pool, you get this. And then we can see that as this goes deeper within the network, this signal might be lost so soon. And in fact, if you look at mass pooling, although it seems better than average pool, but compared to our technique, you see that this can still go further. For example, if you differentiate this, you still get a better feature that goes deeper down within the layer of the network. We tested this technique on CT images, and then we saw that our average pooling performed better than max pooling and average pool. We also developed a custom, cost-effective CNN network where we replaced, you can see here, you have the convolution layer, the batch normalization layer, and then here, traditionally, is always the pooling layer. We replace this pooling, which is usually max pooling or average pool, with average pool. And then the other layers takes the conventional CNN architecture. We saw that our average pooling technique, when we introduce it here, provide better performance. Also, to validate the approach, we consider standard or state-of-the-art models that are available. We use the AlexNet, ResNet, GoogleNet, and then we change or replaced the pooling layers of this network with average pool. We see that when we replace that, the model performs superiorly better. For example, if we consider GoogleNet that uses the max pooling layer, and we replace it with average pool, we get an improved performance here. In fact, a very interesting case is the AlexNet. You can see that the difference, is very significant and is high. And also the same with ResNet. It shows that this average pooling provide a better feature extraction, mechanism compared to max pooling and average pooling. Also, the third thing that we did was published in this journal. Basically, what this study does was to propose a model that used an ensemble technique. And what are we trying to achieve here? For example, in the hospital, lung cancer is basically, diagnosed and treated by, a lot of specialties. You have the radiologists, oncologists, you have the medical physicists, and, a lot of them that come together, for the patient treatment. So we simulate that scenario that if we can have different models coming together to, analyze the data and come up with, a final diagnosis, it might present a better solution than using a single model. And then we tested that approach. We developed, this algorithm flowchart where we have, we use the input, here is the input image, the CT image. We perform image segmentation of the region of interest, and then we pass this into the 3 different models where you see this is the model 1, model 2, model 3. We are trying to simulate, for example, 3 doctors who are analyzing this scan, and then they come up with different, diagnoses. We evaluate each of the diagnoses, and then we assemble them to form a single diagnosis. We evaluated that, for example, in lung cancer detection, we saw that if we look here, the proposed model that combines the 3 models is the deep nodal detect that we proposed, and then these are the results of each of the models. We saw that the ensemble model at every case provided improved performance here, and here. Also, same as when we try to classify the lung cancer, after detection, then we can say, oh, there's lung cancer here, but what type of cancer is it? For example, adenocarcinoma squamous cell, we try to, make the model to classify the data for faster, diagnosis. And then we see that the ensemble model at every instant surpass, the individual models here. Another interesting thing that we observed was that if we augment the data, the performance improved. If we apply segmentation of region of interest, it improved performance also. And here we can see the table of confusion here that shows the confusion matrix for each classification. We compared our study or result with previous studies that have analyzed the same data set that we analyzed. For example, here we saw this study, Karim et al., Suleiman et al., and we see that ours, using this data set, provides or gives the best results. And also for this data set, the chest CT scan data set that we employed to classify lung cancer, to date in literature, our results remains the best, achieved accuracy in this regard, as of today in literature. In conclusion, our study demonstrates that, in fact, we can use a low-cost deep learning model to effectively, diagnose lung cancer using patient CT scan. We also saw that if we utilize ensemble technique, data augmentation, segmentation of region of interest, the performance improved. In the future, we aim to extend this study to other imaging techniques such as the MRI, ultrasound, and X-ray imaging because, for example, MRI and ultrasound, they provide or introduce less ionizing radiation risk to patients compared to CT scan. And what does this imply? The implication means that now, with lower computational cost, local hospitals can develop deep learning models or automated detection and diagnosis system in their hospital, not relying on high performance computing, which a lot of hospitals cannot afford. And also we saw that our method was able to solve the problem of requiring large amount of data to achieve performance. So with limited data in your hospital, we can design a very cost-effective model for you that can run on the normal computer or normal machines, and then we can achieve significant and improved diagnosis. Our code is available in GitHub. You can check it, and it's freely available as long as you cite, the publications. We appreciate the CSIR for their financial support. Here are the references that was used for the presentation, and thank you.
Thank you very much. A very needed study, very interesting. Do we have any questions? Yes, please go ahead.
Hi, thank you for your very interesting study.
Thank you.
I just wanted to ask, how early can your model detect lung cancer? How early on is your model able to detect lung cancer?
You're saying how early?
Early, yeah.
Yeah. For us, study shows that as early as possible, let me use that, because we do not know when the patient presents at the hospital. We based it on the data that we analyzed. So if you have 100 patients presenting, they must have presented at different timelines of the cancer stage. And then for our model to be able to, achieve almost about 99 to 98% detection accuracy, it shows that it can detect the cancer at the earliest stage. And then we saw through our analysis that there are, patterns in the image that are not visible to oncologists or radiologists. We tested that. You give an image to the radiologist, and then they tell you this is a normal scan. And then we put it to our algorithm. It tells us this scan is cancerous. And then we compare with the result of biopsy results, and then we see that it is true. So it means that our model can detect lung cancer earlier than radiologists can. So this actually is what makes the study very interesting, that it can be, practical for hospitals to, have models like this. Although, like I said, if you look at this slide here, it is not replacing the the radiologist. You can see the AI here, it's informing the radiologist. There's a CT scan, it's been processed by our algorithm. The AI tells the radiologist what it sees. Then, within a very quick time, the radiologist can confirm, what the AI have seen. And it makes the job very fast, effective, and then we can achieve more accurate diagnosis, because in the hospital, a lot of the time, false positive are detected, and in fact, even false negative, where there's a patient that comes in, for example, in routine. What they do now, for example, in advanced country, they do routine lung cancer screening, where people who are at risk of lung cancer, for example, who are smoking, they come for that screening, and then they use automated diagnosis system to analyze the data, and they get, good result. A lot of people were diagnosed from, studies like that, for example, in the UK, and it has helped to improve survival, particularly for lung cancer.
Thank you very much. I can take one last question. Yes, please, Prof.
Thanks, Epi, for that presentation. Just the first one, how do you ensure that you're not overfitting that model to reach the ranks of 99% or whatever it is?
Okay, for us to be sure that we didn't overfit the model, we employed several metrics, like I said, sensitivity, accuracy, and AUROC. But the killer method that we used to know was the one that I described in terms of generation capability, where we trained the model on a data that was acquired with an imaging system, and then we evaluated with data that were acquired with a different imaging system of different population. In fact, in 2 different continents. And then we saw that, in fact, the performance was consistent compared to other methods. So that tells us that our model is not actually overfitting the data. It is learning the true representation of what it is to be lung cancer.
Last one. So in all of these models, you use what, 80-20
Pardon?
ratio? In your models, what ratios of train test?
Training, we use 70-30.
70?
Yes.
30.
30 for training and then 30% for test,
Okay.
which is the standard in deep learning mostly, basically.
Okay, thank you very much.
Thank you so
Can we
much.
just give him a round of applause?
Okay, our next presentation is by Toboane Ramolongo, who is exploring the synergy between traditional medicinal knowledge and modern computational science. So her official title is Integrating Indigenous Medicinal Knowledge with Computational Modeling for the Discovery of Antiviral Drugs. Thank you very much.
Good morning. I just need to make sure that I don't forget to breathe. Just looking at the title, you can see that this is a very ambitious project. It is a multi-disciplinary project that touches on indigenous medicinal knowledge. On that alone, you need an understanding of the indigenous uses of the plants, which then it brings in the social, cultural aspects, and then the use of medicinal indigenous plants. Then it brings in bioprospecting and metabolomics. The second thing is the computational modeling for drug discovery. Then it brings in molecular and structural biology, artificial intelligence, and machine learning. And another thing is virology. So you need to be able to understand the intricacies that are associated with the viral life cycle. My name is Toboane Ramolongo. I am a researcher with a PhD in microbiology with expertise in virology. So I am based within the Indigenous Knowledge System program under the Advanced Food and Agriculture cluster within the CSIR. So I will just skip through this, then you'll catch up as I go through the presentation. Just to give a background on IKS, Indigenous Knowledge System is a very broad term, so it touches on the knowledge, the skills, the know-hows, the technologies, the innovations, amongst the others. And this is the knowledge that have developed within the communities for centuries, and it has been passed down from generations to generations. So the communities, they have used this knowledge to adapt so that they can strive within the natural environment. And then this IKS is cross-cutting, and then it finds applications in agriculture, in health, food product preparations, and environmental conservation. So with environmental conservation, it was interesting to find out how they do this. So they use taboos, like let's say for plants, if they see that the plants is starting to be becoming extinct, then they start creating stories against that so that people, they don't use it that much. So I am within the IKS program, so we are mostly focused on medicinal plant species, so we do mostly the validation of the indigenous knowledge so that we can substantiate their claims and also develop products. So the importance of IKS is seen on how from how the public is interested in this field, and that is reflected by the legislations that have been placed to protect the knowledge and also the indigenous genetic resources. So one of the acts or the legislations that is in place in South Africa is the National Environmental Management, specifically the Biodiversity Act. So this act is enacted to protect the indigenous knowledge and also it's enacted to protect living organisms. It can be animals, it can be plants, it can be microorganisms. So if we are doing any activities that's associated with naturally living organisms, then you activate the compliance of the act. So with the IKS, there's this one regulation that emanates from Chapter 6 of the Biodiversity Act. So it is the bioprospecting, access, and benefit sharing. So it says that whenever you do research that associated with the natural resources or with indigenous knowledge, you need to make sure that you are compliant. So when you do research, you go collect a plant, you need to make sure that you notify the Department of Forestry, Fisheries, and the Environment that you're doing this and this with these resources. And then once you come to a space where you want to now patent or you want to commercialize your product, then you activate the commercialization phase permit system. So this part of the commercialization phase has taken us, the IKS team, to the bundles where you find yourself with a suitcase filled with long skirts, because now you need to adjust to the cultural expectation of that area. And this is the last act, so is the Indigenous Knowledge System Act. It has a very long name, which was signed by the President in 2019. So this is a sui generis act which strictly protects IKS. You can see that IKS space is highly regulated. So likely for us at the moment, although this act has been signed to law, it's not yet enforced. So I've mentioned that the indigenous knowledge is passed down from generation to generation. Mostly, you find that this knowledge is living knowledge, which means it's orally transferred from the generation to the generation. So what the GSTI, and in collaboration with the CSIR, what they did many years back, they created this system, the NICMAS, which is, so in this system, it is a recorder system where you find all the information that you need on plant species, and then you find also the communities that are associated with the traditional uses of those plants. So it actually is very useful for us researchers, because then you can go to communities to source this information. So you can actually find all the information regarding the traditional uses of certain plants. But at the moment, it's very restricted, so you can only access certain information, but you cannot access the details of the communities. So it is still an ongoing project where they're still documenting this information across the country. So from this system, we were able to identify 62 plant species that the communities are using to treat respiratory infections. And the other thing when it comes to IKS, let's say with, okay, I'm going to use COVID because then it's more related with RNA viruses. When you have a COVID-19 infection, then you, as a communities, they don't say that we are treating this disease. They are more treating symptoms. So when you work, even from the recordal system, you find information based on symptoms than on the actual disease. And then I mentioned that this project has also the antiviral aspect to it. Then the question is why there's so many diseases, why then you choose viral infections. So WHO, the World Health Organization, time to time, they update that they publicize the list of priority diseases that the research community needs to focus on to develop diagnostics, therapeutics, and vaccines. So this list of pathogens or diseases, they are selected based on their possibilities, the potential to cause the next pandemic. And after the COVID-19 pandemic, then the WHO decided to also add the disease X, which was never in the list. And then the disease X is there to denote that the next pandemic might be caused by a pathogen that we have not encountered before. So if you take a closer look to all these viruses, you find that they are caused by, but these diseases, they are caused by RNA viruses. And then most of them are zoonotic, which means they are transferred from animals to humans. Just to give more of a picture of how often we encounter the spillovers or the emergence of these viruses over time, you can see this is the timeline infograph that shows the spillovers of these viruses from, this is over two decades. You can see that most of them that are here, they are listed under the WHO list. So there's Ebola, CCHV, and the Lassa virus, Zika. So most of them, they are listed as the viruses that still needs the research communities to develop diagnostics or therapeutics. So the one thing also that I want to mention with these viruses, the one thing that's playing a role with them being emerging or re-emerging is the global warming, and also the animals, they're finding themselves being displaced from their natural habitats, and the people are also interacting more with wild animals. Then this assists to the spillovers, the transfer of these viruses to humans. So we really need to prepare ourselves for anything that's still yet to come. So this is more of the strategy that we want to use towards in this project. When it comes to drug discovery, there's a conventional way and also the in silico modern way. So I'm a person who don't like things that takes time. So with a conventional way, you need to have a whole lot of plants that you need to screen, and then you can see down here. So you need to first identify the target, and then you do all the screening and everything in the lab, which is slow. But then with the in silico modeling, it's faster. So I've mentioned that we are using indigenous knowledge. So what's happening with indigenous knowledge, it guides us to select plants, because you don't want to just go out there and take everything and then bring in and work with it. So indigenous knowledge guides us to streamline the plants that we can work with. And then we use the chemical profiling using the LC or MS, LCMS or GCMS to create the profile from the plants. And then so with computational modeling, then you will need the compounds from the plants, and then you also need a target protein. So I'm going to explain a bit more on the target protein, why we chose the specific one that we are working with on this project. So you can see with the in silico modeling, once you have the ligands and then the target, then you can run all this in the model, using different models, of course, and then you can have heat compounds that then you will bring in the lab and then do your toxic cytotoxicity and all that before then you go to in vivo. So it kind of like streamline the process that you need to actually get a therapeutic product. Okay, so I've mentioned that the diseases that are listed by WHO, they are caused by RNA virus. Most of them are caused by RNA viruses. So I remember during the COVID pandemic, one morning we just wake up and then we'll be told, oh, there's another variant that's spreading like fire. And this is because that the RNA viruses, as they make copies, as they replicate, they create mistakes which makes them, which then create new variants. But then also the virus has to have, it has some proteins that it actually needs to survive. So it has to make sure that those proteins, they do not change for it to replicate. So the RNA viruses has this protein, which is the RNA-dependent RNA polymerase. So it helps the virus to replicate. So this is the structure of the RNA-dependent RNAs, and then it has an active site. So that means then when you look for therapeutics, if you can block this site, then the function of this protein won't be effective, and then the virus will not replicate. So, and then I've mentioned that this protein is present in all RNA viruses. So once you have a drug that works against this protein, then you can prevent the infection of all RNA viruses. So that's why we chose this one. So for this study, we plan to follow a very straightforward approach. So this part of the project is something that's still very new to me. I am a virologist, but I'm excited to learn on this part of the project. So we planned to create a database of compounds that are present in the plants that we have chosen. And then from there, we will do a molecular docking analysis with the RNase-dependent RNA polymerase as our target protein and the photocompounds as ligands. And then from there, we follow by doing ADMET analysis. ADMET analysis is just to check the drugableness of the compounds, and then followed by the molecular dynamic simulation, which then determines the stability of the complex, because then the protein and the ligand and the target needs to make a complex so that the protein does not function anymore. So you need to check also the strength of that interaction. So after you do all these things in the computer, you can say that, okay, I have my drug, because then you need to still go back to the in vitro, do your in vitro studies, and then check if this is the true picture that you have within the biological system. So this pretty much summarizes what we need to do. So I have one case study, we did more of a prelim evaluation of this. So we chose this plant, Kigelia africana, is also an indigenous plant. So for this one, the compounds, they're already well documented, because that's another problem with indigenous plants. The knowledge, the information, the data is very limited. But for this one, we're able to get about 152 compounds from the literature. So we did the molecular docking simulation. So another thing with the RNA-dependent RNA polymerase, the source structure of the protein is readily available, and then what you need to do is just to prepare the structure of the protein, and then you clean up your ligands, and then you do the docking. So this is one of the results that we did once we ran the molecular docking. So we were able to find two compounds that showed high docking scores, and the docking scores, they were compared to remdesivir. Remdesivir is a FDA-approved clinical drug that is used against the RNA viruses. So you can see that the compound 1 shows like -11, and then compound 2 a bit lower, but -10, which is almost in the range of remdesivir. So this is the prelim work. So we still need to do the ADMET analysis, molecular dynamic simulation, and then the in vitro studies to confirm if this is the true picture that we get in vitro. So basically, this project, where we want to take this project, we want to prepare ourselves for the next pandemic. So we have rich knowledge of indigenous. We have rich indigenous knowledge and also the diversity of our medicinal plants. So we want to create a database where we can actually have it readily available and also feed back to the NICMAS, the recordal system, so that at least once it gets open to the public, then you can see also the scientific information that's associated with those plants. And then also we want to be able to have a whole process from the database and also from the modeling and the in vitro that we can actually just pull out if then in case of the next pandemic, because then if you have these plants growing, then the communities can just use that to treat whatever virus that you have. And then so with IKS, whatever we do is linked to communities. So that means at the end of the day, if we discover a drug that can be commercial, so that means then the communities that are associated with that, they will also benefit. So that will be our end impact. So we have a product that also impact the community. Thank you.
Thank you very much. Very interesting. I actually appreciated you spending time on the X because there is that sensitive line that we need to toe between research and indigenous knowledge. I'll be taking a few questions. Oops, sorry. That's my time. It doesn't want to stop. Any questions? Please go ahead.
Okay. Thank you, Dawani. That was very interesting. I'm your colleague, but every time I hear that, it's very interesting to see how IKS can be even integrated with like computer systems. So on the microbiology side, you mentioned something about docking. Is that when you bind like the interaction that you have to limit the growth of the protein?
Yes, yes. So, like with the plan is that the way some therapeutic works. So you have you have a virus. So virus produces a whole lot of proteins when it replicates. So you just choose which one you want to focus on, which is then to the target protein, and then you have your compounds. So you want a compound that will be able to bind to that protein, so that that protein doesn't do its function anymore. So if it can't function, that means the virus will not be able to replicate anymore. So that's the interaction that we need to establish.
Okay. So how does the binding happen? Like, is there a certain criteria for the compounds to actually bind with the protein? Like, how do you know that? Or is it a trial and error kind of, or you have to?
So for this one, I'm not sure if I'm going to answer that well, but we are using Schrödinger Maestro. So that's the program that is developed for drug discovery. So it does have all, so you can actually set it to the way that you want. Like you can set the limit if then you don't want the interaction. If you want it to be in this range, then you can set it up. So that's within the model, which I am not very clear.
Oh, okay. Yeah. No, thank you.
I'll just take two more questions. So I'll start with the lady there, and then Paul after. Then
Thank you for such a great talk. I wanted to find out, since you mentioned that you targeted the active site, did you also maybe consider blind docking, especially if there is allosteric inhibition?
You see, can I have a conversation with you after this? Yes, that's all I can say. Yes, I still need a bit of more help when it comes to the modeling. Yes.
Unjani Tobani?
Hi.
Ngiyaphila. My question will practically be a tad bit off course.
Sorry, say that again?
My question will be a tad bit off course, but it's
Okay.
related to what it is that you're doing.
Okay.
You are specializing on IKS. So when you were gathering the information, are you necessarily only basing this on anecdotal evidence? Do you include, or rather, did you, in the process of gathering information, consult, say, maybe the traditional healers organizations? Because if I'm not mistaken, some of the cures that you guys are actually either discovering or attempting to prove their validity has already been discovered by the traditional healers, more so because it's indigenous knowledge.
Yeah, yeah.
Did you by any chance consider or contemplate roping them in? Secondly, would it also be possible for you guys to integrate what they already have into what you are developing to actually facilitate for either the comparison thereof of the results that they get that have been tried and tested by time, vice versa, the modern day sciences and methods of medication that are being conducted and used today? Thank you.
Yeah, that's a very
10 seconds to answer.
No, it's actually not, because when we do community engagements, it's mostly when we are at the commercial phase. So that means at that point we've already done the research and everything. And the most of the concern that they raise is that they would have loved for us to consult them at the beginning so that they can be part of the process. But the challenging thing with the AKS is that most of this knowledge is oral, and then at the beginning of research, you just maybe find from a publication that there's this the traditional use of these plants for this and that, and then you do the scientific validation. So you will only be required to have the beneficiary agreement when you do the commercialization phase. That means then you have to go and then consult those people to have the agreement. But at the beginning of the project, like I said, the plant species that I have identified for this project, I found them from the NICMAS, from the recordal system. And then already on that system, you can only see limited information. Like the communities, the GSTI is still withholding that, so you cannot get. So you can only get that these plants that is traditionally used for this and that. And then we will do the research, and then if then we find information that is like we confirm that is true, and then you can actually have a product. That's when you will be required to have that agreement with the knowledge holders. But at the beginning, it's very difficult. But it's something they will prefer for us to do. Yeah.
Thank you very much. Paul, can I just ask you to take your question offline? Apologies for that, but we are running over. Oh, Pinda, are you feeling sorry for him? Yes, so in the morning, so can you please give her... In the morning, we were told that this is a nice and friendly environment, so please forgive us a little bit for running over time. So our next speaker is Matapelo Motau, whose research is focused on the nuclear latifolia, and the official title of the presentation is Evaluating the Effect of Nuclear Latifolia Plant Extract Against the Candida albicans virulence. Virulence, yeah.
Good day, ladies and gentlemen. My name is Matapelo Motsau. I'm a master's student from the University of the Witwatersrand, and I will be presenting my research titled Evaluating the Effect of Nuclear Latifoliae Against Candida virulence. Okay, so Candida albicans is a commensal microorganism that is found in the skin, in the oral cavity, in the gastrointestinal tract, as well as the vaginal tract. It exists in different morphological sites. So there's a transition in morphology of Candida. As we can see with our picture at the top, we have the yeast form, the pseudohyphae, and the hyphae form. So the pathogenicity of Candida albicans is also influenced by a number of virulence factors, such as adherence, biofilm formation, the formation of germ tube, and also the production of hydrolytic enzyme. And due to this, Candida albicans is able to cause an infection that is known as oral candidiasis in the oral cavity. Sorry about that. Okay. So with my research, because of Candida can be found at various sites in healthy individuals, my research focused mostly on the oral cavity. So what happens when a person is infected with Candida albicans? The microorganism attaches to the epithelial cells of the host. Once that happens, this process is known as adherence. Once this happens, biofilm is able to form, and in the same length also, we see the formation of hyphae. So the importance of hyphae in the infection is that the hyphae is able to allow the microorganism to actually penetrate into the epithelial cell of the host. Once the organism has penetrated in the mucosal membrane of the host, we now see the production of hydrolytic enzyme. And these hydrolytic enzymes are namely phospholipase and proteinase. So the importance of these hydrolytic enzymes is that they degrade the phospholipids and the proteins that are essential for the survival of the human cell of the host. So once this has happened, once the infection, the microorganism has invaded the epithelial cells, it is now an infectious disease. Now the infection is progressing, and we see in most people who are infected with oral candidiasis is that we see oral thrush in their mouth. So the common name for oral candidiasis is oral thrush. This is because of the overgrowth of candida in the mouth. And usually this happens in people who are immunocompromised, people who have weakened immune system. And we also see it mostly in children, because we know that children obviously have weaker immune system compared to adults. So currently, there's quite a limited number of antifungals that are present for just overall the treatment of fungi. And these antifungals are mainly the azole and the prolines, right? But the challenge with the present antifungals that are present is the emergence of drug resistance. So yes, we have drugs that are available, but they are limited. And also, something that I can also add is that when compared to bacteria and viruses, fungi has quite a limited number of antifungals that are already available in the market when compared to other microorganisms. So now, in addition to that, we add that there's an emergence of drug resistance. Hence, there's a need for the development of new therapeutics for oral candidiasis. This then leads our research to actually look into medicinal plants. The reason why we're looking at medicinal plants is actually because of they have been used previously, traditionally, by Asian folks, And also, it's said that they do have bioactive compounds that are able to treat different kinds of diseases. Hence, we are looking at medicinal plants. One of the plants that we actually looked at in the studies is nuclear latifolia plant extract, which has been used traditionally to treat dental cavities and oral sepsis. And also research has shown the potential of this plant used at the different parts of the plants have been used to test its potential in antibacterial, as an antifungal, and also as an antiviral. So what has not been done actually with this plant is to test its potential as an antivirulence. So the reason why we are looking at the antivirulence is because of, as I mentioned at the beginning, is that Candida albicans is a commensal microorganism. So we still need it in the body, though it is an opportunistic pathogen, but we don't want to kill it, we want to inhibit its virulence. So hence the need to actually be able to inhibit the virulence that occur when the infection does occur. So this leads me to the aim of my study, which was to determine the antimicrobial effect and the antivirulence potential of nuclear latifolium against Candida albicans. The objectives were to determine the antifungal effect as well as the antivirulence effect of the mentioned virulences, and also investigate the effect of the plant extract on the virulence genes of this microorganism. 5 clinical strains of Candida albicans and an ATC strain was used, and all the experiments were repeated in triplicates. Ethical clearance was obtained for the use of the fungi of the Candida albicans, and as well as the use of the epithelial cells that were donated. For the methods, the micro broth dilution was used to determine the MIC, which is the inhibitory effect, and also the fungicidal effect of the organism. Once we got the MIC, then the MIC that was obtained was further used to determine actually the effect of the plant extract on the adherence of the fungi to the epithelial cell, the formation of biofilm using the crystal violet method, and germ tube formation, the production of proteinase using BSA agar because it's able to produce nitrogen, phospholipase production using the egg yolk, and also the gene expression was determined using qPCR. For my results, the MIC that was obtained was 0.16 milligrams per mil, and the MFC was 0.63 milligrams per mil. The MIC that was then obtained, which is the inhibitory effect of the plant extract, was further then used, as mentioned, to test the virulence factors. So it was used at those concentrations that are here. For the adherence assay, the percentage reduction that was seen was up to 85%, and at the lowest concentration which was used, which was 0.04 milligrams per mil, it was 73%. And the significance of the plant extract being able to inhibit adherence is that adherence is actually the first step of the infection. So if ever the plant extract is able to actually inhibit the first step of the infection, it means that there won't be any progression of the disease. For the formation of biofilm, this was done at 6 hours. So according to literature, biofilm of Candida albicans is able to form by 6 hours. We see that the biofilm has formed. So we saw a percentage reduction of up to 50%, and at 6 hours, and also at 24 hours, we saw a percentage reduction of up to 40%. And these were at the higher concentration of the MIC that was 0.16 milligrams per mil. So with biofilm, it is the reason also behind drug resistance is because of biofilm. So we can see from the results that it becomes very tricky to actually deal with the progression of the infection once the biofilm has formed, because of biofilm actually have extracellular matrix, and these extracellular matrix protects them from the antimicrobials that might be given to the host. So it is quite difficult to deal with biofilms in various microorganisms. It's not only in Candida. Another thing that was observed is the percentage reduction of germ tube. So as I've said previously that Candida is able to exist in different morphologies. So the germ tube, which is the hyphae, it's actually the most pathogenic form of Candida, and it therefore assists it in the penetration to the epithelial cells of the host. So we saw a percentage reduction of up to 51%, and so if ever the reduction of the hyphae is, if ever like the hyphae is reduced, it means that there will be less available hyphae that are in the host that will be able to penetrate into the cells. So hence it was important for us to see a reduction in germ tube. Sorry. Okay. And then with proteinase, there was no effect that was seen or observed when we tested the proteinase. And the proteinase are actually enzymes that degrade the proteins in the cell membrane. So this shows that once the microorganism has invaded in the host, it becomes quite challenging to deal with it, because of now the infection has already progressed. So that we could see that there was no, the plant extract was not able to prevent the degradation of the host cell, because of it had already invaded the host. For phospholipase, we also saw that there was a low and moderate activity of the low and moderate activity that was observed of the reduction of this plant. And the PZ value are actually we are measuring the precipitation zone of the effect of the thing, of Candida albicans. So we could see that at 0.76, that's actually a low enzyme activity that was observed. So when we can see that it is able to block the secretion of phospholipase. So phospholipase, when compared to proteinase, it's able to be blocked. Because of proteinase, we could see that it was not able to be blocked by the plant extract. On the gene expression assay, we saw a downregulation on ALS3, which is a gene that is responsible for adhesion, and HWP1, which is a gene that is responsible for hyphae formation, PLB1, which is responsible for phospholipase, and the sub-genes that are the proteinase. So the reason also behind us doing the gene expression, it was to validate the results that we obtained in the other assays, that is it really, when we check the genes, the virulent genes, is it really having that same effect? And we could see that it was able to downregulate all the genes that are expressed. And one thing that we could see was that on proteinase, one of the proteinase genes, which is SEB3, there was quite an upregulation when compared to the other SEB genes. It's because of there are a number of SEB genes that do take that play a role in this microorganism. So the upregulation might be that there's literature that still states that sometimes when you introduce antimicrobacterial into a thingy, when you introduce an antifungal into Candida, what it does is that it actually initiates, the microorganism becomes more excited. So instead of it actually being suppressed, it actually goes up. Hence, we can see the upregulation in SEB3. To conclude, from my results, we can see that N. latifolia is able to inhibit Candida albicans. It has an antifungal activity. And we also saw that its potential in antivirulence effect in adherens, phospholipase, hyphae, and biofilm. And we could see this, we can confirm this by the downregulation of the virulent genes. So what can be done is that we can now start doing more investigations in terms of the cytotoxic effect of nuclear latifolia, because of what we would not want is this plant extract to be actually toxic, because of we know that fungi is actually a eukaryotic, and also mammals, humans actually are also eukaryotic. So what could be done further is to check its cytotoxic effect on human cell lines, and also to characterize the bioactive compounds that may be found in the plant, because of I did not go through the process of characterizing the bioactive compounds that are actually in the plant. And once this is done, then the plant extract can be taken into further and being tested in animal models to see how effective it can be, so that it can be able to help individuals that do have oral candidiasis, because of most of the people who have oral candidiasis and are immunocompromised find it difficult to eat and to chew. And if this is the case, they won't be able to take their medication for whatever that they might be struggling with outside of oral candidiasis. These are my references, and I would like to thank my supervisors as well as the CSIR for giving me the opportunity. Thank you very much.
Thank you. So I'll be taking 2 questions. Do we have any questions? Yes, please.
Thanks for that presentation. May you please go to slide 2? I mean, not slide 2. Yeah, figure 2.
I don't know what I did there.
Can you maybe start with the question while she finds the slide?
I want to reference the figure. There, in any case, we have those concentrations at the bottom, but controlled. There's concentrations up to 0.6. But you can't get to the figure.
Sorry?
You can't reach the figure.
Which figure? Sorry.
Figure 2. Not slide 2, figure 2. Yeah, there. Yeah, even there. So in that trend, 0.16, what would have happened if you had gone further? Would that decrease?
If what? I don't understand the question.
On your y-axis,
Okay.
Okay.
You've got the concentration is at 0.06, what? I can't quite read that thing. But on the x-axis, you've got a concentration that is increasing.
Yes.
The bar graphs are also decreasing. So I'm asking if you had gone further,
Okay, yes.
would those bar graphs continue that trend? In other words, there is a negative linear actually.
I really I don't know whether I not getting the question, but you're saying if ever I was going downwards, meaning
If you had continued increasing that concentration.
Yes, oh, okay, now I get it. So the more we increase the concentration, the more effective it would be, if I'm getting what
So
you're
is
saying.
there data demonstrating what you're saying? Have you gone further than what you've reported?
No.
One more question.
Thank you, ma'am. Nice work that you're doing. It's very relevant. So this is just a question of more of if I was an entrepreneur, how would your work give me like a market advantage?
How would my work give you a market advantage? Okay, so because of oral candidiasis affects people that are mostly immunocompromised, right? So let's say that we have a lot of people that are sick because of once the infection has progressed, there are high chances of the patient actually dying, right? So if ever we are able to prevent these people from actually getting to that stage, and these people can be people that are employed in different industries. So imagine now you're having a lot of your people, because of whatever diseases that they might be facing, and then oral candidiasis occurs as an opportunistic pathogen. So it means that now you will be losing, a person, an employer might be losing people because of people are actually sick in the hospital because of there's no treatment for this infection and stuff like that. So as a marketer, you know that people will need to buy this medication so that they prevent the progression of the disease. I don't know if.
No, very much. Thank you. Thank you.
Thank you very much. Can we just give a round of applause? Okay, so we are almost at the end. Our final speaker is Halaletso Otibeng, whose work highlights the pharmacological richness of local flora and also demonstrates the power of in silico profiling in modern natural product research. So the title for her talk is Computational Metabolomic Profiling of Anti-Venom and Anti-Inflammatory South African Medical Plants. Please come through.
Okay. Let me tell you and just stand up straight. Okay. How it works down arrow for next, up previous, this is your printer. Okay, got it. Thanks. Okay, good day, ladies and gentlemen. As introduced, my name is Halaletso Otibeng. I'm a PhD candidate at the University of Johannesburg under the supervision of Dr. Fidel Tugzimana, Dr. Zamani Cele, and Prof. Justin van der Hooft. And today I'll be briefly sharing on my ongoing project, which focuses on the use of computational metabolomics to profile or elucidate anti-inflammatory and anti-venom South African medicinal plants. Now, medicinal plants have been used for centuries for the treatment and management of a wide array of disease conditions as well as ailments, ranging from colds and flus to skin diseases, malaria, cancer, amongst many others. And South Africa is actually one of the countries with a long history of the use of medicinal plants, as well as a rich biodiversity of these plants, with over or about 60% of the South African population relying on medicinal plants as their primary health care option. Now, the therapeutic and health benefits of medicinal plants can be attributed to the bioactive compounds that they possess. Now, these are also termed plant-derived natural products, and they are continuously being explored for the development of new drugs. Now, there has been indeed multiple drugs that have been developed from plant-derived natural products. A typical example is artemisinin, which is isolated from the plant Artemisia annua and is popularly known for its use as an anti-malarial drug. Now, previously, there had been a significant decline in interest in natural product research, particularly from medicinal plants, and this was owing to several reasons, such as the perceived long discovery and development process, complexity in the synthesis process, as well as the high frequency of re-isolation of known compounds and difficulties in elucidating the chemistries of these traditionally used plants. However, the past few years have seen increasing or a resurgence of drug development from plant-derived natural products, and this is driven by advances in the technologies as well as the development of strategies that help us accelerate this discovery of plant-derived natural products for drug discovery purposes. Now, South Africa is well positioned on a global landscape owing to its rich biodiversity of these traditionally used medicinal plants. Two examples of these are Melianthus comossus and Hypoxis hemerocalidea, which are the plants that are under study. And these, although they have many uses, we are mostly interested in the uses for the treatment and management of skin diseases and conditions, as well as snake bites. Now, Hypoxis hemerocalidea has also been known, or is popularly known, as African potato, and this is due to the fact that the shape of its bulb mimics that of a potato. Now, the two plants were selected, number one, based on claims of the anti-inflammatory and anti-venom activities or benefits that they possess, as well as based on their availability and accessibility. Now, with regards to how exactly these plants are used on traditional medicine practices, the roots and the leaves of the plants are normally boiled, and then the decoctions or the pastes are then applied on wounds, swellings, and burns in order to promote their healing. Baths with the herbs have also been said to relieve joint pains, while the roots of Melianthus comossus have been used in the treatment and management of snake bites. Now, despite the many uses of these plants, there is, however, a limitation with regards to knowledge on the phytochemistry of the plants. Now, this speaks to what is in these plants, as well as the underlying biochemistry, which speaks to the specialized metabolisms of the plants, and more simply put, which compounds in the plants are responsible for the benefits that we see and experience. Now, omics sciences such as metabolomics offers unique opportunities for us to be able to comprehensively profile the chemistries of these plants and better understand the underlying biochemistry of these plants, and this is an advantage towards natural product research for drug discovery and development purposes. Okay, so what is metabolomics? So it is a system biology discipline that involves the qualitative and the quantitative analysis of the metabolome. Now, the metabolome is basically all the metabolites that are in a biological system of interest. That can be a cell, a tissue, or an organism as a whole. Now, this is just to show some of the applications of metabolomics in natural product research, as well as some of the work that has already been done in this regard. Okay, now despite the maturation of the field of metabolomics over the past decades, and despite some of the technological advancements that have been made regarding the analytical platforms that are used, identifying or annotating these metabolites still remains a great bottleneck. Now, this then limits the biological insights or information that we can generate from such studies. Typically, only about 1.8% of spectra that is generated from metabolomic studies can be annotated, meaning we can put names to these detected chemical signals, while the rest or majority of them still remain unknown, falling under what is called the chemical dark matter. Now, computational tools or computational metabolomics has then been proposed as a solution to help us overcome these limitations of metabolite annotation and identification. Now, given the artificial era that we are currently in, the past few years have seen efforts from the scientific community to develop as well as apply several computational tools and strategies that will help us decode these chemistries of medicinal plants even further, be it at a structural, substructural, or even at class level, helping us know more than we would using the traditional approaches. Now, this is just to show some of the computational tools that we are exploring and applying in this particular study. Okay, so the study had three main aims. However, for the purpose of the presentation, I will be focusing on the first one, which looked at elucidating the metabolic landscapes of the two plants under study, Melianthus commosus and Hypoxis hemerocalidea. Experimentally, the following steps were followed. So the plants were purchased from a nursery in Mulder's Drift, and harvesting of the leaves and the roots of the plants were done. So we focused specifically on these two tissues because they are the ones that are majorly used in traditional medicine practices. So now to extract metabolites from these tissues, we used methanol as well as followed the hot water extraction method. Now the second was done in order to mimic what is done in traditional medicine settings. As I mentioned, how these are used, the roots and the leaves of the plants would be boiled, and then the paste would be applied on the wounds and swellings to promote their healing. This was then followed by analysis of these extracts on liquid chromatography mass spectrometry platforms, and the generated data was then analyzed, processed using various platforms. Now, to annotate or put names to these chemical signals that we detect, we explored various computational tools such as molecular networking. This was then followed by biological interpretation. Okay, so I hope you can see. So making use of these various computational tools, we're able to tap into or annotate quite a lot of metabolites from the metabolomes of these two plants, from the leaves of the plants as well as the roots of the plants. Now, one trend or pattern that one can see is that we were able to annotate more of the flavonoids from the leaf samples in comparison to the root samples, while looking at the lipids, more of the lipids were annotated from the root in comparison to the leaf sample. So this is just one way to illustrate how the use of these tools can actually help us tap into the chemistries or the underexplored chemistries of these traditionally used medicinal plants. Okay, so to put into context or show how exactly these tools help, so over here we made use of the tool referred to as molecular networking. Now how this works is that it groups compounds that are similar in their spectral information or their fragmentation patterns together, and this then same principle is then followed in the annotation step. What this means is that compounds that are similar in structure would be grouped together, and then how this helps is that if I'm able to identify at least one or two of these nodes, I can use that information to then extrapolate and postulate the identities of other unknowns in the same cluster. A typical example, so over here a node, the circle refers to a query spectra, which represents a metabolite of interest, and these two were matched to reference spectra in GNPS house libraries and putatively annotated as being the flavonoids nicotinflorin as well as rutin. So this is one way in which the use of computational tools such as molecular networking can help in terms of propagating metabolite annotation, reducing the re-isolation of the same compounds, as well as increasing then the probability of us discovering novel bioactive compounds that can be candidates for drug discovery. Okay, so another advantage of this tool, feature-based molecular networking, is that it allows us to visually represent the metabolome that we extracted. So you're able to visualize all the extracted metabolites that you got from your leaf or your root samples, and also it integrates or incorporates relative quantification information, helping us then to also see the differential distribution of different metabolites across the leaves of the two plants. So taking flavonoids for an example, we saw that chimephil retinoside, luteolin glucoside, and isoquercetin, as well as their related compounds, were more abundant or more predominant in the leaves of Melianthus comossus in comparison to the African potato. Whereas if you look at the flavonoids such as vicinine 2, corimboside, and saponin, these in this case were more predominant in the African potato leaves in comparison to the Melianthus comossus leaves. Now what this shows is that it speaks to the differences in the chemistries of the two plants that we are working with. Another interesting observation we made was that we annotated a lot of the glycosylated flavonoids. So to then zoom further into these, we created sub-networks of these glycosylation patterns. So over here, the shape of the node correlates to the type of flavonoid, while the colors of the nodes correlate to the sugar that is attached to these flavonoids. So we are able to see how there is quite an interesting glycosylation patterns of the flavonoids in these plants, and with the glucose conjugated flavonoids being the more abundant in this regard. We also looked at how these flavonoids were distributed across the leaves of the two plants, where we saw that chimephil in its A-glycan form, that is when it's not attached to a sugar, was more predominant in the leaves of Melianthus comossus, while when we looked at the glycosylated forms, we saw that chimephil 3 retinoside, in this case, was more predominant in African potato leaves, while chimephil 3 beta-xylose was more predominant in the leaves of Melianthus comossus. So this speaks to differences in the specialized metabolisms of the two plants under study, which can be translated to variations in how they are applied in traditional medicine practices. So this study, to conclude, this study contributes to global efforts to look back into medicinal plants as sources of natural products that we can use for the development of new drugs. And South Africa, with its rich biodiversity of the plants as well as the rich traditional medicine knowledge, stands to really give a lot in this regard. So this project is contributing towards efforts of also bridging the gap between traditional medicinal knowledge as well as modern science, and this can speak to policies, conservations, as well as strategies to implement some of these in the healthcare system. So from the generated maps, the use of the tools helped us to gain better insights with regarding to the phytochemistries of the two plants that we're working with. And we saw that they spanned various chemical classes from tannins to flavonoids to ACA compounds, amongst many others. Moreover, we looked into the differential distribution of the metabolites across the leaves of the plant, speaking to differences in the chemistries of the plant. We also looked at the glycosylation patterns of these flavonoids and their distribution thereof, speaking to the specialized metabolisms of the plants that we are working with. And although not shown, another step that we did was perform computational modeling. So that is molecular docking as well as molecular dynamic simulations in order to predict the activities of selected metabolites. As part of the ongoing work, we are currently busy with the in vitro anti-inflammatory and anti-venom biological assays, and also exploring more of these computational tools to help us dig deeper into the chemistries of these two plants. So, yeah, this brings me to the conclusion of my presentation. These are my references. I'd like to acknowledge the following individuals and institutions and thank the CSIR for this opportunity to share my work. And last but not least, I'd like to acknowledge the Tukizimana Research Lab. Thank you.
Very interesting talk. Questions? Please go ahead.
Thank you, ma'am. Nice research and
Oh.
nice presentation. So let's say you had more resources like time, finances, and more hands to help you with your research. What would you have improved?
What I would have improved would be, okay, so I am limited with regards to the tools that I can use based on my limited coding and programming knowledge. So if I could get expertise in that regard, that will help me also now expand further. And yeah, and with the efforts towards the fractionation and the biological assays, that will really help us get to the more active fractions that we can then take further to NMR to structurally characterize the structures and identify these compounds that can be used as candidates then for drug discovery.
Thank you. Thank you very much.
Any more questions?
Thank you very much for the presentation. Very interesting. In terms of, maybe if you can go back to the slide where you are showing the processes, the analysis, HPLC stuff.
The steps, the methodology?
Yeah.
Oh, okay. I'm going the wrong way. Sorry.
Oh, yes. So okay, just looking at that, you mentioned that after your extraction and then you do your analysis on your LCMS and then the data mining, is there a specific software
that I use?
Yeah.
Yes, so depending on where you analyze, there are some vendor-specific platforms. For instance, with the Waters instrument, you'd use the Waters Connect software, but there's also open source platforms such as MZMine3 that helps us with processing these, and also it can even annotate as well for you as well, so yeah.
Okay, so will this be like sort of a foundation? Because if you go through your LCMS and then you have your data mining, will it be like a precondition for your isolation so that you don't, because with your LCMS data, you wouldn't know what chemicals to isolate. So will that be like a shorter way to get to the type of compounds that you would isolate going further?
Yes and no. So in our case, we do not use it for that. So the first thing that we wanted to do was explore the phytochemistry or the metabolome in its entirety, regardless of what's bioactive and what's not. And from that, also using the in silico tools as well as from the assays, would then narrow down our search to those that are specifically active against the venom and the, yeah.
Okay, thanks.
Sure.
Yeah, thank you for the presentation. I think my question, it might be related to us in regards to the data mining.
Okay.
I just want to know which techniques did you use, and also for your molecular networking, how long does it actually take with your current tools?
Okay, so for the data mining and processing, like I said, I use the MZMine3, so you can download it online. There's also MS-Dial, which is another option that you can use. And for molecular networking, so it's an online-based tool called GNPS. So what you do is that after you process, you get two files. That's your quantitative information file as well as the one that has your spectral information. Then those are the files that you use to generate these networks. And with these, with the current updates with the tools, it actually takes very little time, in less than 30 minutes. Well, depending on how big your data set is, you can actually generate the network quickly.
So basically, is it like machine learning?
Yes, there's also some tools that incorporate machine learning and deep learning.
Okay.
Yeah.
In this case, which one did you actually use?
We use okay, so remember from that list of the many tools? So the results that I showed were from this one, which does not use machine learning. However, MS2 Query does incorporate some deep learning aspects.
Thank you. Thank you very much.
So I'm going to take one last question. Go ahead, Devon.
Thank you so much for the presentation. During my presentation, I touched on the National Environmental Management Act. So I see your project has kind of like triggered that. How compliant is your project?
Yeah, to be quite honest. You know, when you presented, I was like, I hope she's not going to ask me that. Here, I'm actually not sure, but that is something to look into. We did not go outside to the traditional healers as well to try and get information from them. But we can speak further.
Yeah. Even the research itself, it triggers the act.
Oh, yes, because you can't just like take a plant and do some analysis on it. Yes, I do understand.
Okay, thank you. Can you just
So that leads us to the conclusion of the session. I would like to sincerely thank our speakers and all of you for participating and making it a really nice and engaging session. And yeah, can we just give all the speakers the last round of applause? And then please feel free now to go and have some lunch.